fourth lecture in the History of Medicine series. It gives me great pleasure, and it's really a great honor to have Peter Agre today as our speaker. Peter Agre shared the 2003 Nobel Prize in Chemistry with Roderick McKinnon for his discovery of aquaporins. And these are the channel proteins that allow water to cross the cell membrane. He completed his clinical training at Case Western Hospitals at Cleveland and the University of North Carolina. And then he returned to Hopkins in 1981. And here, and this is where we were discussing the role of serendipity as well as the role of mentors in these discoveries, in that his discovery of aquaporins was serendipitous. And he was able to show that a particular Rh protein, which he thought was a contaminant, indeed happened to be the channel. And I will not tell the story because he's here to tell us himself. And the Nobel Prize enabled Dr. Agre to take his research and scientific interest to new directions. So he felt that his laboratory work had taken him away from his main interest in serving mankind and medicine. And so he has now turned his focus back to third world diseases, and he's now serves as the director of the Malaria Institute at Johns Hopkins, where he has applied his knowledge to the study of the malarial parasite and the Anopheles mosquito, which both express aquaporins. So most importantly, I think the hook that got him to accept this invitation is the fact that he's passionate about encouraging young people to go into science. And so I hope that this talk serves all those purposes. Welcome, Dr. Agre.
Thank you, Sarah. I have to apologize. I have a voice problem. I have Parkinson's disease and sometimes don't project my voice well. So if that's the case, please wave your hands or let me know and I'll try to do a better job. I'm very grateful for the chance to be here. Sarah and I have been working on this for several months to get the schedules right, and her request was very special. She wanted a historical talk. Although I have to say the field that I've been working in for a long time, water transport, thank you. I need all the help I can get. The field was fairly negligible, and so there'll be a lot of Hopkins events that I'll present, and I hope you'll have some questions. So way back in a previous century, this young man with a backpack came to Johns Hopkins with an interest in global health. I don't mean to scare the young people, but that was me. Time is cruel. It's relentless. But I came with a lot of enthusiasm and the hope that through medical science, something important in terms of global health would emerge. And I worked in a laboratory in the basic sciences, also at the Bayview campus, on the problem of infectious diarrhea, cholera. And I have to tell you, if you work on diarrheal diseases, you can't expect that to enhance your social life. It's sort of one of the parts of science that is not so glamorous, but is of great consequence to the developing world. So certainly cholera is still a major health problem. And of course, the problem with cholera, this horrible diarrheal disease, is the maldistribution of water. Water is often mentioned in the lay press, problems with the global water, the Katrina hurricane, the melting of the Greenland ice cap. There are a lot of global water issues that are oftentimes brought to our attention. But water is also important in biology. And just with global water, where disequilibrium can lead to dire consequences, the same can happen in terms of water and biology. So the history was, I'm going to summarize it. Back in the 19th century, when Claude Bernard talked about the sea interior, the milieu of our bodies being an aqueous sea, he was not aware of cells. Rudolf Virchow established the cell theory, and it was in the 1920s where Evert Gordel discovered that the cells were encapsulated with a lipid bilayer, which was semi-permeable and assumed to be partially permeated by water. And this cartoon reflects that. The bilayer on the upper left, as predicted by Gorder and Grendel, is slowly perfused by water. Water will diffuse into the cell and out of the cell. It's a temperature-dependent process. It's a slow process, but it occurs in all of our cells. Now, I think the clairvoyants were a small group of biophysicists led by Arthur K. Solomon at Harvard Medical School and others, George Benga in Romania, Guillermo Wittenberry in Venezuela, a group of scientists who could not accept that simple diffusion could explain the massive water permeability seen in some tissues, such as renal tubules, secretory glands, and red cells. And while they were committed to the notion that there were special channels that must exist, none of the techniques for identifying those channels were productive. But they did very precise experiments, and they established that the presumed water channels present in these highly permeated tissues had a great selectivity for water. It was not permeated even by protons, the hydronium ion. And the movement through these putative water channels was believed to be directed by osmotic gradients. So secretory glands could release water, renal epithelium could absorb water, but they didn't know the mechanism. I think a very important experiment was undertaken and reported in 1970 in a rather obscure biochemical journal. And the report by Robert Macy and Robert Farmer at the University of California, Berkeley, was quite interesting. They were studying red cell water permeability just because of basic interest in biophysics. And they found that some chemicals, notably mercuric chloride, would stop water transport across red cell membranes. And when they treated the membranes with chemical reducing agents, they could restore water transport. They could turn it off and on, and concluded there must be an aqueous pore, which Solomon predicted, with free sulfhydryl. So that's, by and large, the history. Again, they were stuck because they didn't know what the channel was, and by and large, the scientific public didn't believe there were channels. I went through an entire course in physiology here as a Hopkins medical student, and we studied everything about the kidney except for how the water moved through the kidney. So, as Sarah mentioned, serendipity has a large role in medical science. And in our case, it was clearly an unplanned accident. We were studying the rhesus blood group antigens, an important red cell antigen. After the ABH system, the RH is the most important clinically. And back in the early 1980s, when I was joined the faculty here in the Department of Medicine, it was not clear what RH was made of. But we were able to isolate a 32 kilodalton polypeptide from surface labeled red cells that turns out to be the core polypeptide of the rhesus blood group antigens. And if you look at this solid SES polyacrylamide gel stained with silver reagent, you'll see that in addition to the 32 kilodalton polypeptide, there's a slightly smaller polypeptide of 28 kilodaltons, which we incorrectly assumed to be a proteolytic fragment. So this is a humble beginning for a new field, a contaminant which was misidentified. But there were some curious behaviors of this 28 kilodalton polypeptide. It was present in all red cells of different species. It was extremely abundant, but it stained extremely poorly with Kumasi and other reagents. So basically no one had seen it before because of its chemical behavior, but it was a very abundant polypeptide. And so the emerging revolution in molecular genetics was occurring. I did a sabbatical at the Carnegie Laboratory at the Homewood campus to learn how to do cloning and sequencing. And we cloned out the cDNA encoding this polypeptide and found a very interesting gene product. The deduced topology indicated that it resided within the lipid membrane, and it spanned the bilayer multiple times with a putative repeat. The first 3 bilayer spanning domains are repeated in the back half of the molecule. And there were some homologs in the genetics database. This is in the early 1990s. The genetics database was pretty small back then. But there were related proteins from very interesting, diverse origins. A protein referred to as major intrinsic protein from the lens of eye from beef cattle, a polypeptide expressed in the brains of insects, Drosophila, Melanogaster, and others, a factor expressed in bacteria involved in glycerol metabolism, and a large series of fragments of genes from various plant tissues. So we were quite interested in this, but really didn't have a clue what the function of the protein was. And I'm not exaggerating that. We'd purified it, we'd cloned it, we were ready to do some experiments, but didn't have any clear ideas of what to do. So this is where I'd like to introduce another subject, that's called the work-life balance. Something that is now talked about in graduate schools and medical schools, but it was never mentioned once in the 1970s. The idea is you work as hard as you can and hope your family survives was the method. But my wife, who worked at Hopkins for Diane Griffin in the neurovirology laboratories, when we got married and the children arrived, decided we would absolutely take a family vacation every summer at a national park. No work, just camping with the kids. And the kids loved this. We went to Yellowstone, we went to the Sequoia, Yosemite, the Grand Canyon. And after a few years, Mary said, let's ask the kids which national park they want to go to next year. And so we did, and they all yelled Disney World, which is not a national park. But families are not obligate democracies, so we went to the Everglades. We then went to Disney World, and on our way back to Baltimore, stopped in Chapel Hill, North Carolina, where I spent 3 years as a hematology fellow at the University Hospital. And Mary and the kids were visiting friends in Chapel Hill, and I talked to a number of colleagues, including this man, John Parker. John was a very wonderful hematologist, a superb clinician, who had a small physiology lab studying membrane transport. When I described this 28 kilodalton polypeptide and the notion that it must have some important transport function or some membrane function, John thought for a moment. He said, you know, red cells, renal tubules, this protein is present in 2 of the most water-permeable tissues in mammalian biology, plant tissues also. And then he said, have you considered this may be the long-sought water channel that Solomon and others have been looking for? And it had never occurred to me. It was a new concept, water channel. I'd never heard the words. But it was based on John and his insight that he steered me to Bill Gugino in the physiology department. Bill was a junior faculty back then. We both were. He's now the chairman of the physiology department. And it was really a collaboration with Bill Gugino. And I think that's what makes Hopkins special. People actually collaborate with each other. They help young scientists. And so he expressed his new complementary RNA in Xenopus laevis oocytes and observed them, and the oocytes looked the same. Control oocyte on the left, test oocyte on the right. There was no obvious difference. They were cultured for 3 days, but when they were transferred together to distilled water, there was a huge difference. The control oocyte is now swollen negligibly, but the test oocyte is swollen and exploded and produced much pandemonium in the laboratory. This was Greg Preston, the postdoc who did some of these early studies. And I took a snapshot of Greg actually 3 years after his first report. He was still celebrating. So I think we're probably not like the general public. I think we generally do things that are useful, and we should pursue our fantasies and celebrate when something works. But there became a large amount of interest in the transport world, and so we quickly decided we were going to have to collaborate with specialized groups if we're going to make progress on this, because the discovery of this protein, which we termed Aquaporin 1, the first water channel protein, was of large interest. And structural biology, of course, has now blossomed. It informs us much about the function of molecules. Just as Louis K. Sullivan, the famous architect, said, form always follows function. The same is true in biology. So we teamed up with Andreas Engel on the right from the Biocentrum in Basel, Switzerland, and Yoshinori Fujiyoshi from Kyoto University, and by the technique of membrane crystallography, solved the structure at an atomic level resolution. And as you can see on the left panel, a subunit viewed from the top, the extracellular surface, there's a single aqueous pore down through the center, about 3 angstroms in diameter. And on the right panel, a cross-section, you see a single channel. It meanders a little bit from side to side, connecting the extracellular surface of the membrane with the intercellular surface. And this cartoon, developed by David Cousino, explains how water, which exists in bulk solution in the extracellular space and intracellular space, water can cross the membrane through an aqueous pore of about 3 angstroms in diameter, which is precisely the molecular diameter of water. And there are fixed residues blocking the movement of charged solutes. And in the very center, there are 2 shared asparagines in the 2 repeats, which fold from the intracellular surface and the extracellular surface right into the middle of the membrane, where a single water molecule is isolated for a billionth of a second, preventing proton transport. And this is David Cousino. He's now a radiation oncology faculty member at Harvard Medical School. And the structure at the very center of the channel shows the tight fit, a space-filling representation of a single water molecule surrounded by special residues that prevent the movement of solutes and other substances. And if you look at the channel, I'm not having the best luck with this pointer. There's a cysteine. If you imagine the channel being a clock, at 4 o'clock, there's a cysteine, which is the mercury inhibitory site that Macy had described. So we teamed up with others to establish the function of this protein. The nephrologists had the greatest interest in water transport, because our body water homeostasis is determined by our kidneys, as is the pH. And we were fortunate to be introduced to Søren Nielsen, a young scientist at the University of Aarhus, Denmark. He was 29 years old when we met and first started working together. And Søren was a world-class microscopist with a special interest in kidney. And we knew that our protein was very abundant in kidney. And if you look at the left panel, there's a representation of a single nephron unit. Our kidneys are formed of 1 million or so nephrons. You can see the glomerulus where filtration occurs, and in green I've highlighted the proximal convoluted tubule and descending thin limb of the loop of Henle. So the primary urine filtered through the glomerulus moves through the proximal nephron. And on the right panel you can see the osmotic water permeability. This is a compilation of studies from multiple different investigators, but the proximal nephron, highlighted in green, is very permeable to water. The next segment, the ascending thin limb and thick limb, are virtually impermeable to water. And then the urine is dumped into the collecting ducts, which are interesting because water permeability is regulated by antidiuretic hormone, also known as vasopressin. And Søren's micrographs were superb. Here's one from a rat kidney. On the lower left, there's a proximal tubule. Can't point. You'll have to just take my word for it. The lumen is marked with a P, and it's surrounded by a brush border which is intensely stained with a specific antibody to Aquaporin 1. And each of the epithelia of the lateral membranes and basal membranes are also decorated with the antibody. The collecting ducts are devoid of staining, marked with a C. So the collecting ducts, where final concentration occurs, must have another homologue. So we believed, and that turned out to be the case. And by immunogold labeling, you can see an intense level of expression at the apical brush border, where Aquaporin 1 is extremely abundant. So it's is not present in the cell body. So it's not cycling back and forth from the cytoplasm up to the surface. It remains fixed at the apical surface where it's constitutively active for water transport. And how this functions, demonstrated in this cartoon, our kidneys will filter approximately 200 liters of plasma every day, an enormous volume. And the water is reabsorbed in the proximal nephron, and as well as in the collecting ducts, through the aquaporin proteins. And as you can see, the lumen above, a water molecule passing through the aquaporin 1 molecules, and the protein is tetrameric. It will pass through the cytoplasm and exit through the lateral and the basal membranes of the epithelial cell in a transcellular movement. And the movement of water is driven by the osmotic gradient created by salt and solute transporters. So there's a small standing osmotic gradient. And this allows the proximal nephron to reabsorb at least 90% of the filtrate. That's a huge volume. Now, in the human genetics program here at Hopkins, Chol-So Moon was a graduate student from Korea. He did his medical degree at Seoul National University. And as part of his thesis project, he localized the structural gene for aquaporin 1 to the human chromosome 7. This was before the genome had been solved. And it was actually a collaboration with Connie Griffin here in the Cancer Center that localized the structural gene to the short arm of chromosome 7, where interestingly a blood group antigen referred to as colton happened to have been localized. So there are hundreds of blood group antigens, but they're actually comprised of about 25 different families. So the genetic locus coinciding with our structural gene was a strong clue that they may be the very same molecule, and that turned out to be the case. Polymorphism in an extracellular location of the aquaporin 1 molecule is a colton A-B polymorphism. But the power of the blood group referencing system is enormous because they reference blood group antigens in patients all over the world. And there were a half a dozen individuals in different countries who've been identified with colton null. They had no colton antigens. So we were able to get some blood from these colton null individuals and found that they had knockout mutations in the gene encoding aquaporin 1. And here is a photograph of a retired school teacher from the south of France with her permission. She had developed antibodies to the colton antigens during pregnancy. She was colton null. And we looked at her phenotype, and this was orchestrated by Landon King, who was at that time a postdoctoral fellow. He's now the executive vice dean. He's running this university. But he started out as a house officer and a clinical fellow, and he established with great certainty that there was a defect, a marked defect in the urine concentration of the colton null, the aquaporin 1 null individuals. After overnight thirsting, all of us will concentrate our urine to about 1,000 milliosmolars. This individual and the two others we studied could only concentrate a little bit. It stayed just above physiological tonicity. And while they were subclinically affected, after overnight thirsting, they were still releasing large volumes of dilute urine. So the phenotype would actually be very severe if an individual was restricted from having free access to water. The protein is also extremely abundant in capillary endothelia. And in another collaboration, Landon led with Robert Brown from the anesthesia department using a high-resolution CT scanning technique of the lung developed by a young Hopkins faculty member named Elias Zerhouni. That name may be familiar to some of you. Again, an eminent scientist, leader of academic medicine who came to Hopkins to do research. And using this technique, they imaged deep in the lung and the distal bronchiolar level. The structures at baseline, you can see these are the normal individual. You see the lumen of bronchiole, the L represents the airspace. Notice the wall thickness and an adjacent venule. Then the same individual was infused with 3 liters of physiological saline. And what you should see is that the venule now has increased in surface area because it has become distended. And we found that with the null individuals as well as the controls. But if you look closely at the CT images, the lumen of the bronchiole now is greatly thickened in all normal individuals, but that did not occur in the null individuals. They could not move water into and out of the venules rapidly. And probably the reason the phenotype is so rare is because of the importance of the alveolar and bronchiolar water absorption of amniotic fluid at the time of birth. So it turns out to have a very significant phenotype. So laboratories around the world interested in transport found this irresistible, and with homology cloning techniques rapidly cloned a large number of aquaporin DNAs from different sources. The mammalian genomes have 13 different aquaporins, and they're comprised of two different subsets, the water-selected channels, the classical aquaporins, and the aquaglyceroporins, which are permeated by water plus glycerol. And while humans have 13 different genes, other species have different numbers, but every living organism has one or the other, or one of each. And so I'm going to talk about some of these other aquaporins and aquaglyceroporins. And this is work of many different laboratories. So as I mentioned earlier in the talk, the collecting ducts don't have aquaporin 1, and it's clear that this is where regulated water permeability is known to reside. By homology cloning, Sasaki and his colleagues in Tokyo isolated a cDNA, which we now refer to as aquaporin 2, expressed in the isolated renal collecting ducts. And if you look at the electron micrograph, the upper panel, you see that this immunogold staining is largely not at the cell surface, but over intracellular vesicles. So the lower panel on the left shows an isolated collecting duct stained with aquaporin 2 selective antibodies after the tubules have been pulsed with vasopressin at physiological levels. So by exocytosis now, the upper surface, the lumen surface, has water channels that have permeated it, allowing water to enter the epithelium. It then exits through lateral or basal membranes through other aquaporins, aquaporin 3 or 4, depending on the level of the nephron. Now, inherited defects in the aquaporin 2 are extremely rare, but they're very profound. Children who are homozygous for knockout mutations in the aquaporin 2 were identified first in the Netherlands. And these children had to drink up to 20 liters of water a day to maintain fluid balance because they could not concentrate their urine. Acquired defects turn out to be extremely common, with overexpression found in basically all states of fluid retention, congestive heart failure, for example, and underexpression in all forms of enuresis, including bedwetting. So one by one, each of these was studied by different groups, our group included. The protein in the lens referred to as major intrinsic protein, we refer to as aquaporin 0 because it had been identified already. It turns out that mutations in the DNA encoding aquaporin 0 lead to congenital cataracts appearing in small children. That was work of Masato Yasui. He was a postdoctoral fellow here at Johns Hopkins. Aquaporin 4 is present in a number of tissues, including brain. And in a collaboration with our colleagues in Norway, Ole Petter Ottersen, who's presently the rector of Karolinska in Sweden, but he was a neuroscientist at the University of Oslo back when we worked with him, and established the location of the aquaporin 4 water channel in brain. Now, capillaries throughout our bodies are rather fragile. You twist your ankle playing tennis or jogging, it'll swell up, and in a week or so, the swelling will subside. But our brains cannot tolerate influx of water. Our cranium, which is rigid, protects the brain, but if a brain injury occurs, any swelling will be accompanied by compression of adjacent brain tissue. So on the left, I borrowed from Eric Kandel's book, a diagram of a brain capillary. You can see it's surrounded by a basement membrane and astroglial end feet. And these astroglial end feet form the so-called blood-brain barrier. And if you look on the right, Otterson's group decorated the brain sections. You can see an abundance of staining. If you look at panel B in the lower right in particular, the protein is present right below the basement membrane in a polarized distribution. This predicted the protein would have a functional role in the manifestations of brain edema. And continuing our work here at Hopkins with Dick Trason and his colleagues in the neuroradiology unit, we studied both normal mice and mutant mice, the mutant mice having targeted genetic defects in the proteins bracketing Aquaporin 4 at the proper membrane. And this is led by Mahmoud Amiri, a very interesting young scientist, a European neuroscientific leader who started his career as a refugee in a refugee camp in Pakistan. He was adopted by the Norwegian Social Services, where his talents emerged. And what Mahmoud has discovered is that all normal mice, when receiving a defined brain injury, a occlusion of the middle cerebral artery, and reperfusion, all the normal mice will have infarct and edema. When the mice are sacrificed, the red appears. The rose staining is a viable tissue. The pale represents infarcted and edematous tissue. You can see that the mutant mice, which have Aquaporin 4 but at the wrong membrane, they're protected from the brain injury. Now, brain injury after stroke, head trauma, or brain tumor, the brain edema is a very wicked, nefarious source of much suffering. This is an individual, I know this well, this was my wife 4 years ago, when she appeared to have a fairly abrupt course of meningioma. If you look at her MRI in the upper left, you can see a very large meningioma. Hard to imagine someone could walk around with a tumor like that and feel well and do well. But there's a threshold where you can't compensate anymore. And if you look at the upper right, when the MRI images are weighted for water density, you can see that the tumor is surrounded by a massive degree of brain edema. Now, the lower panels show that the tumor was mercifully removed entirely by Henry Brem, very skillful neurosurgeon we were fortunate to have here at Johns Hopkins. But the brain edema persisted, and it persisted for weeks. This is a problem. Now, I should have a slide saying I have a disclosure. I hope to have a disclosure. There's a biotech firm which we're working with that has developed inhibitors to Aquaporin 4, which may block the appearance of brain edema. The firm is entitled Aeromics, and we should know soon if the clinical trials are productive. Another member of the family, Aquaporin 5, this was cloned out by Surabhi Raina, a young postdoctoral fellow from University of Delhi. She's now a professor at Syracuse University. And when Surabhi was here, she cloned out the aquaporin from secretory glands. It sent the last membrane water crosses during the generation of tears, sweat, and saliva. Now, in Denmark, Lenny Nasum studied Aquaporin 5 with a knockout mouse prepared by Anil Menon and his colleagues at the University of Cincinnati. So if you look at the panel on the left, that's a normal mouse paw coated with starch and iodine, injected with pilocarpine. The sweat released from the sweat glands has amylase and degrades the starch, and you see the blue dots. These are functional sweat glands. On the right, the paw of an Aquaporin 5 null mouse, which has sweat glands, but they're hypofunctional. They don't sweat, they don't release amylase. Now, rodents live in the cool shadows during the day, they're nocturnal. But humans labor in the hot sun. The ability to sweat in extreme circumstances will be a matter of life and death. Now, the summer of 2003, Western Europe had the most profound heat wave in recorded history. The temperatures hovered at or above 100 degrees for 2 weeks in a row in Paris. And the public health officials were aware that there were 15,000 unexpected deaths. There was a massive heat wave leading to deaths of older individuals. And as we age, presumably, our physiological pathways decline in efficiency, and the ability to sweat, sense thirst, and concentrate our urine probably all declined, and the individuals didn't survive. So I think, while the pathways may be important in many physiological situations, at times of stress, their functions become essential. Now, briefly, I'd like to talk about other members of the family, which we refer to as the aquaglyceroporins. I had been on the faculty quite a while. I had never thought for one moment about glycerol transport. I don't think there was anything in our medical school lectures about glycerol transport, but there was a protein studied in bacteria known as GLP-F, which turns out to be a homolog of the aquaporins, and the structures shown here were reported by Robert Stroud at the University of California, San Francisco, and Andreas Engel, our colleague in Switzerland. And he's juxtaposed the pore lining residues from a water channel, which are shaded in the darker colors, and the corresponding residues in the glycerol transporter. And as you can see, they largely correspond, except for in the lower right level, lower right corner of the water channel. My Aquaporin 5 is not sweating enough to let me move the cursor. There's a histidine that I'd like you to observe. That's a positive charge which repels protons. And in the glycerol transporter, it's replaced by glycine, a smaller residue, which allows the histidine, the pore to be a larger structural magnitude, because it allows the phenylalanine 200 to drop like a trap door. I'm not doing a very good job of describing this, but you may get the idea. So glycerol transport turns out to be important in skin. This is a series of skin sections prepared by Johan Ogren, who did a sabbatical in our laboratory. And the aquaglyceroporin 3 is very abundant in the basal levels of skin. Now, it didn't take long for the beauty industry to get interested in this. One summer, I was contacted by some executives from the Christian Dior company who wanted to come visit. Now, I don't know about your laboratory, maybe Steve's lab, but most laboratories don't have a lot of visitors from the Christian Dior executive offices. So I was highly suspicious. And they told me about a new product that would come on the market, which contains some natural substances which lead to upregulation, increased expression of aquaglyceroporin 3 in sun-exposed skin. The implication being, if you buy this expensive skin cream and use it, you'll look like a model. I'm not personally convinced. And I have to say, I have no financial ties to Christian Dior. Something which my wife reminds me frequently was probably not a good decision. They offered some nice support, but I didn't think it was proper. This was the back cover of the Marie Claire beauty magazine. And if you read French, you'll see that it notes profound hydration, spectacular results, and the Nobel Prize in Chemistry in 2003. I showed this to my mother back in Minnesota. My mother was, at that time, about 80 years old. Was a farm girl. She never went to the university. But she saw this. She said, "Peter, I think you're finally doing something useful." Imagine that. I'll tell you about two other aquaglyceroporins briefly. Aquaglyceroporin 7, which is expressed in fat in adipocytes, and aquaglyceroporin 9 in liver. So we have a new disease in North America and Europe. It's called obesity. 60 years ago when I was a kid, obesity was uncommon in the schools. Maybe one child would be obese, and of course the poor youngster would be ridiculed by his classmates. But now it's very common. About a third of elementary school children are obese, and almost half are overweight. And it's because we're hardwired to efficiently store extra calories. Our progenitors were so stressed, and even into the last century, the Native Americans and First Nation in Canada, remote locations during the winter to survive starvation. They would store extra calories as triglycerides and fat. And during fasting and starvation, of course, the triglycerides are degraded to glycerol and fatty acids. And the glycerol is released from fat through aquaglyceroporin 7. It's taken up by the liver through aquaglyceroporin 9, and by fasting, the level of 9 is increased in the liver, and the glycerol is then efficiently transported into the liver, where it's remanufactured into glucose. So we can maintain our blood glucose levels even to the late stages of fasting and starvation. And this is work of Jen Carberry, who's now a faculty member down at Duke University. Now, you never know what you're going to hear next about a new research project. I was contacted by Barry Rosen from the Wayne State University, who was interested in studying arsenite in yeast. And he made some yeast arsenite null strains, and when he transfected them with the aquaglyceroporins, he could measure increased arsenate uptake. So as it turns out, the aquaglyceroporins are very efficient in transporting arsenite. Now, in eastern India and Bangladesh, where cholera is very abundant, the government has put in tube wells to draw out the groundwater. And unfortunately, in many settings, the groundwater is heavily contaminated with arsenic, leading to a massive arsenic poisoning. The World Health Organization predicts 40 million people are drinking arsenic-toxic water every day. And of course, there's a marked increase in the appearance of hepatocellular carcinoma. And our colleague Søren Nielsen in Denmark, who prepared an aquaglyceroporin 9 null mouse, and you can see the liver sections, the wild type on the left, the null on the right. When they were injected or fed arsenate, they could not excrete it in the feces. Of course, they succumbed to arsenic poisoning. So probably the evolutionary importance of the aquaglyceroporins, in liver at least, is to again defend against arsenic poisoning. And all organisms have at least one member of the aquaporins. Here's a slide I borrowed from Ralph Kaldenhoff from the University of Würzburg in Germany. And the wild type Arabidopsis, a small mustard plant, you can see maintains a arborization, lush arborization, which is fully hydrated with a rather skimpy rootlet complex. He then prepared genetically modified Arabidopsis, in which the rootlet aquaporins of one family, there are actually 35 different aquaporin families in Arabidopsis, but one family, when the plants underexpress them, the plant compensates for the reduced water permeability by sending out a thick arborization of rootlets. So on the left, you can see the plant is compensated. Now, plants compete for water. In the dry season, in the Savannahs of Africa, the rhinoceroses and the elephants and the elands will all migrate to different water holes. But the plants that remain are competing for water, and probably the aquaporin expressions in plants will be manipulated to help create drought-resistant crops. If there's no water, of course, all the crops die, but the competition for the last remaining water is pretty intense. So I'm going to summarize the talk to this point. I've talked about the aquaporins, the free permeation with water and very selective for water. I've talked about the glycerol transport of the aquaglyceroporins, the structural models to support them, and listed just some of the multiple clinical disorders where these proteins are involved. But doing something about it is much more difficult. And so I'm not sure that this knowledge has been so valuable yet in terms of human medical practice. So I can't talk about this without digressing a little bit about the Nobel distraction. It was 15 years ago, the phone went off early in the morning. A very pleasant voice on the other end, a Swedish accent. This is an important telephone call from Stockholm for Professor Peter Agre. Are you Professor Agre? And I said, I sure am. And they informed me I would share the Nobel Prize in Chemistry. Now, I was not a very good chemistry student in high school. My dad, who was a chemistry professor at a small Norwegian college that I ultimately attended, he thought chemistry was the most important subject. And probably to rebel, I let my chemistry lessons go on unfulfilled and pulled a D in high school chemistry. So I'm thinking about this as I'm hearing from the Nobel Committee. Is this a good idea to tell them that I got a D in high school chemistry? I thought otherwise. And then they informed me that in 15 minutes there'd be a press conference and I should get ready for my day. I'm now thinking about my high school chemistry teacher aspirating his cornflakes when he hears the news that his worst student gets the Nobel Prize in chemistry. I sprint to the shower, and my wife Mary, always organized, always thoughtful, calls my mother back in Minnesota. Dad had died 8 years earlier, and wakes her up. It's before 5 in the morning, Central Time, and she informs her that I'll share the prize in chemistry. And mother, the farm girl, thinks for a minute. She says, well, Mary, that's very nice, but don't let this go to his head. We're still hoping he'll do something useful. And by the time I got to the lab here at Hopkins in the morning, it had gone to the heads of the young people. Here's the lab group at that time. These young people came from 8 different countries to work in the lab, and they were on the 3rd bottle of champagne by the time I got in. It was amazing. President Brody came to see me. And until that moment, I had no idea that we were such close personal friends. And I went to the office, and the voicemail was jammed with requests from the New York Times and Lair NewsHour. And then there was a very familiar voice. One of our colleagues from Norway had called. And he was very excited. He left a message. He said, Peter, we've just heard the news. It's unbelievable. And then there's a pause. Then he comes back on and says, no, it's believable. We believe it. And Dr. Zinkum from pediatrics took this snapshot of the Wells Discount Liquor Store on York Road that morning, where instead of announcing the price of Heineken, I have to say the implication that I'm their best customer is a bit of an exaggeration. But you really have no control over the level of celebration. So here I am with my family on the stage after the ceremony, and it was delightful. It was a bit overwhelming, but I think it's clear, at least to me, that the prize is delightful for the individual, but it really is in recognition of the entire field of work. And the rules of Alfred Nobel's will, 1, 2, or 3 scientists can share it, but there's very little work that goes on in laboratories where 1, 2, or 3 scientists have done everything. So I think we should really see this as a great form of PR for what we do, because there's so much great science. And of course, this last, it's still October, I guess, 3 weeks ago when James Allison received the prize, it was clearly a prize that's well deserved. There's so much great work going on. At that point, we had been working on malaria. I had initially come to Hopkins to work in global health, not to become a physiologist or a nephrologist. And we were working on malaria. And the aquaglyceroporin 3 in red cells, it's not expressed in high levels. It turns out to be quite important in the manifestations of malaria. And this is work we did in collaboration with Nirbi Kumar and Dominique Promeneur, who were here at Hopkins at the time. For the parasite to enter a red cell and synthesize glycerolipids, the uptake of glycerol from the plasma through the plasma membrane aquaglyceroporin, AQP3, as well as the parasites, aquaglyceroporins, is essential. So if you look in the near, just left of the center of this micrograph of red blood cells, you see a single red cell infected by a single parasite, a malaria parasite. And that parasite will grow and divide eventually to 32 daughter cells And if you look at the far right in the center, there's a schizon, a red cell where the hemoglobin has been degraded and there are 32 daughter cells. When that lyses, it'll infect 32 new cells, and so it's a logarithmic expansion. 2 cycles is 32 times 32, that's 1,000. 4 cycles, 1,000,000. So in rapid succession, individuals infected with malaria will have hundreds of billions of parasites streaming through their circulatory system, causing horrific fevers and end organ damage. It's a terrible disease. And Dominique's studying strains of mouse malaria, Plasmodium berghei, which she knocked out the aquaglyceroporin. The appearance of the parasite is delayed, and if you look at the far right panel, the mice will have a longer survival, but they still succumb to malaria. So we've assumed that this is not a good drug target because it doesn't totally knock out the parasite. But that's research. The mosquitoes, the Anopheles, which spread malaria, also have 8 different aquaporin genes. Connie Liu and Jason Raskin, when they were here at Hopkins, were studying these. And these allow the Anopheles to move water and survive drought, low humidity, and high temperatures. It also appears that the midgut, where the malaria parasite invades the mosquito, depends on one of the aquaglyceroporins. So when I had the opportunity of switching careers to become the director of the Malaria Research Institute 12 years ago, I was eager to do that because I thought this is something I had always wanted to work on. And it's hard to imagine that this 19th century disease, malaria, is still so prevalent in the world. At least 200 million people will be infected with malaria this year. And about 500,000 children will die. And as you can see from the world malaria map, the greatest abundance of malaria is in Africa. And of course, death in the countryside is very common. People bury their own, and it's a disease of children. So Johns Hopkins Malaria Research Institute has a wonderful field station in Zambia, organized by Philip Tuma. His photograph is shown in the far right. He was a pediatrician trained here at Johns Hopkins, a chief resident in pediatrics. He grew up in Zambia, and has returned and has dedicated his life to caring for the children of Zambia and is leading the malaria research efforts. So the little boys that I just showed you that live in the hamlets outside of our field station are, by rural African standards, fairly wealthy. Their parents take good care of them, they have bed nets, and if they should develop malaria, they'll be treated. But not very far away are impoverished families where the protection is suboptimal, and this little boy was brought to the field station in Mancha, where we are doing our research in Como with cerebral malaria, near death. And Phil saved his life, but as you can see, he has a dysconjugate gaze. He sustained so much damage to his visual cortex that he's permanently blinded. And it's the aquaporin 4 and the perivascular end feet, which are part of the problem in the cerebral edema and cerebral malaria. So the Malaria Research Unit has both laboratory work ongoing in the School of Public Health and in the School of Medicine, as well as field activities. And by just studying the fields, epidemiology, and treating individuals, looking for asymptomatic individuals and treating them, the burden of the disease has disappeared rapidly. You see this panel of bar graphs. Now with the past 18 years, admissions of children to the Mancha Hospital every year, about 1,500 malaria cases with up to 100 deaths. In 2004, the introduction of the artemisinin in combination, a miracle drug for which Tu Youyou won the Nobel Prize in 2011. Ancient Chinese medicine reintroduced is amazingly effective. And everybody was treated and the survival was greatly improved. And then, of course, due to stock outages, the inability to maintain the necessary drug stores in the clinics. The epidemic resurged in 2006, and reintroduction of the artemisinin as well as the insecticide-treated bed nets has led to a great decline, about a 95% decline in the the prevalence of the disease. But it never goes to zero. There are always a few cases that appear, and whether these are cryptic cases in the countryside or individuals who bring the parasites in from distant places, we don't know. We're working on that. And here's some of our field team from the Matcha research site. And so in closing, I'd like to just sort of tie this together loosely by sharing something that one of our postdocs from China taught me. The Mandarin for crisis, Wei Ji, the Chinese speakers will know. It's not one character, it's two characters. Wei, a time of danger, Ji, a time of opportunity. And the dangers are real, whether we're talking about prostate cancer, drug-resistant tuberculosis, or malaria. The real dangers out there. The opportunity that those of us in laboratories and at universities have to do something about it is very special. And with that, I'd like to challenge all of the young people here to stick with the opportunity. Because the years go by quickly. And as we look back on our careers later on, I think it's nice to feel that you made some difference in some research project. But in doing so, probably made friends and colleagues with scientists from all over the world. And together we'll do great things. So with that, let me thank you for the invitation, and I hope there's time for some questions. I'll do my best to answer them. Thank you. Sarah.
So you showed very nicely that over the years, say a 1 in 10 death, 1 in 15 has been reduced to really 1,500 has come down to 35 incidents of malaria, and then there were 3 deaths. But what I see is that there isn't a reduction in the proportion of deaths. And what is it? What is causing that?
Yeah, why most will survive, and some won't. It's probably based on a lot of factors. It may be the nutritional status of the individual, other comorbidities. It's not altogether clear. When someone gets malaria for the very first time, it's a severe problem, a life or death problem. When individuals have had malaria and survived, eventually they develop a partial immunity. And so throughout most of Africa, there'll be few precautions taken by adults, and they won't take prophylactic medicines. If they get malaria, it's like having a bad cold, and their immunity will kick in and eliminate it or keep it at a very low level. Next time you're taking a taxi cab and you have a driver from Nigeria or Ethiopia, ask the driver if they've ever had malaria, and they'll laugh. Of course I've had malaria. Everybody in my country has had malaria. But if they go back after working in the US for 5 or 8 years, the immunity will have subsided, and they'll get malaria again, and it'll be very bad. So the notion that the immunity could lead to a good vaccine is, of course, something being pursued very actively, but so far the vaccines have been partial and temporary. John.
Peter, there's a rumor going around, and it was related to the day you were informed that you had won the Nobel Prize, and it's a quote. I want you to either confirm it or not. Someone was asking you what the most important attribute was, and you said always bringing donuts to the working group. Is that quote true?
Well, I don't recall ever saying that, and for the most part I avoid donuts. But I think Solomon Snyder recognized that his lectures got much better grades from the students when he served donuts. And Saul is brilliant. Maybe he said that.
Well, people don't know it. Peter was the director of the Savannah Molecular Medicine graduate program. And the incredible thing about it was Peter's always had an incredible enthusiasm for students. And it's when I go back and I think about what's made Hopkins great is the realization that when someone comes here, we treat them as colleagues. And I want to congratulate you because you've done that ever since you were not only a student, but a fellow, and then later on as a faculty. So it's one of your incredible strengths. Thank
Well,
you.
you're so kind, John. Thank you. Yes, please.
study several years ago that talked about when cells move and they advance a pseudopodia, that aquaporin is in that membrane and it allows the influx of water so the pseudopodia can expand and extend out.
Yes.
And that it plays a role in migration. I wondered, has that body of literature expanded to migration of cancer cells or other cellular movement?
Well, you ask a good question. There are now tens of thousands of papers on aquaporins. I surely haven't read them all. But the idea of pseudopodia having increased water uptake makes sense. The null people, and now we have aquaporin null people with null, that knocked out aquaporin 1, I talked about them. They don't seem to have any problem with phagocytosis or infectious diseases. So maybe the proteins can cross-cover for one another. Or maybe it's one of these phenomena that at times of stress, it may be a difference which influences survival. I think the baseline diffusion of water is sufficient to maintain hydration of most of our cells. And even the aquaporin 1 null individuals who lack the protein in the choroid plexus where cerebrospinal fluid is synthesized don't seem to have any problems with cerebrospinal fluid. But if they had a spinal injury where they were leaking cerebrospinal fluid, they may not be able to keep up. So I think that's a simplistic answer. But it may well be that nature has designed systems for times of great stress. At that point, it makes a big difference. Thank you. So quiet. Well, thank you. I appreciate it.
Thank you. If I may ask Dr. Balin to come forward and please beg for your patience for a few more minutes while we give him the award. Thanks a lot.
Well, Sarah gave me the honor of doing this for you, which I am pleased to do. I'll make just a couple of comments, and this is to the audience, and especially the young people in the audience, about what we just heard. First, let me congratulate you on your incredible career. But I think what's so scintillating about what you've done is the mesh between the basic science, the translation, and what this has produced for society. I don't think you will ever hear a better outlining of that paradigm. And I congratulate you mostly for that. It's incredible, and it illustrates why we are scientists, and this integration now, for everything the most basic it is, it seems to influence our world in the way that you've just shown. And so we cannot give you another Nobel Prize. We've got that. There is a remembrance here. And Sarah wants me to remove it, which, let me do it with the box. I'll show it to him,
Okay.
and then he can subsequently remove it. But we want to thank you for taking the time to do this and coming here to give your story to us. I gave the first one. I know it's not a trivial effort to go back and put all this together,
right.
even your own career. And it's humbling because, as you said, we stand on a lot of people's shoulders always, and a lot of other great minds. But this is yours in commemoration for your having done this.
Very heavy.
Yes.
And,
If you can just move out a bit.
congratulations,
Yeah, thanks a
and
lot.
thank everybody for coming.
Thank you.